Hi, everyone. This is Physical Principles of Quantum Biology, Chapter 8, Ultraweak Photon Emission and Cell Processes. This is the first chapter of the second part of Physical Principles of Quantum Biology, which will be released by World Scientific in August 2026. So if you are excited, you can pre-order your copy now. As I mentioned, we will be beginning part 2 today on ultra-weak photon emission and cell processes. But before we continue, let's just do a quick recap of part 1, quantum physics of the living world. Einstein said that one can best feel in dealing with living things how primitive physics still is, and I agree. But we have a lot of physics that we can use in studying living processes. In part 1, we considered atoms as they're made of protons, neutrons, and electrons, and those combine to form molecules, conjugated organic and aromatic molecules. We looked at radical pair reactions, as are found in spin chemistry, photoreceptors, which of course capture photons of light. We considered phonons. Those are quanta of vibrations in matter, and vibronic interactions, which combine electronic and vibrational excitations into quasi-particles known as, I guess you'd call them vibrons. Collective interactions, such as superradiance. We thought about non-Hermitian open quantum systems theory, and at that point, the role of quantum coherence and the interplay between coherence and decoherence come into play. We thought a bit about the quantum classical correspondence, and of course, the long-standing question about trivial versus non-trivial effects in biology. But I hope if you've been watching the lecture series so far, you're convinced that indeed living systems exhibit a high degree of complexity at the quantum and molecular level, as discussed by Schrödinger going back to the 1940s. And the high complexity that we see at the quantum scale really precludes the assumption that quantum mechanics are trivial at that level, or that somehow classical mechanics can fully describe living systems at that level. And if I haven't conveyed it already, quantum effects in biology include photosynthesis, vision, cell respiration, proton tunneling mechanisms that we see in many enzymatic reactions, including concerted proton-electron transfers, where electrons and protons transfer in sync, a magnetoreception, according to the radical pair mechanism or other similar mechanisms. Photoactivated DNA repair, ultra-weak photon emissions, which we'll talk about today, electron bifurcation reactions, chiral-induced spin selectivity in chiral proteins, and as we will consider later on in part 2, electrodynamic molecular forces and strong electronic correlations. So, beginning part 2, coherent quantum effects in biology, I've selected another quote from Einstein, which says that the supreme goal of all theory is to make the irreducible basic elements as simple and as few as possible without having to surrender the adequate representation of a single datum of experience. Now, this is another way of saying that the theory should be as simple as possible, but no simpler. A principle also known as Occam's razor. We want the simplest theory that we can use to describe the phenomena that we see, but it can't be too simple or it won't be adequate. And so, with that in mind, we're going to explore ultra-weak photon emissions. We're going to go on to consider electromagnetic oscillations and then chemical dynamics in living systems, and consider the way that electromagnetic oscillations and other electromagnetic effects can actually influence chemical processes and ultimately produce a level of control in biology. Then we'll go on to consider molecular fluctuations, solvent effects, and dispersion, things like van der Waals interactions. Finally, multi-scale modeling of biomolecular systems, and then some of the most challenging systems to model where there are strong quantum correlations that cannot be accommodated by semi-classical approximations at all. So, for some historical context, of course, light has long been known to influence biology. Back to ancient times, the therapeutic use of sunlight predates the recorded history of medicine, and yet a mechanistic understanding of the interaction of light and matter really only emerged beginning in 1900 with Planck's discovery of what is now called the Planck light spectrum, and then furthermore in 1905 with Einstein's explanation of the photoelectric effect. And these are now still two pillars of quantum theory that have reframed light as quantized energy that are furthermore confirmed by fundamental effects like spontaneous emission of photons and the Lamb shift, just to name a couple more for the physicists out there. Light is an essential environmental factor. It powers photosynthesis, regulates circadian rhythms and metabolic rates, and it modulates cell activities, including growth and gene expression in living systems. Now, what's interesting is that the formal study of photobiology was only defined in the post-quantum mechanics era. So this is truly a new development, even in the history of science. Now, pioneers of phototherapy and heliotherapy include Niels Bohr and Auguste Rayleigh. Bohr's 1903 Nobel Prize-winning work on phototherapy treated lupus vulgaris and smallpox with specific colors of light. Rayleigh established the first heliotherapy clinic in the same year, applying high-altitude sun exposure to tuberculosis in this protocol, which was later adopted worldwide. In 1932, Bohr gave a famous speech to the light therapists. So interested were they in the potential for quantum mechanics to explain the medical effects that they were seeing. At the International Congress on Light Therapy in 1932, Neils Bohr extended quantum principles to life sciences, explicitly challenging the idea of a pure physical-chemical reductionism as a way to explain life. And this inspired, for example, Max Delbruck to pivot from physics to biology, and he later went on to win a Nobel Prize himself. Now, we've shown some people here from the turn of the century basking in the sun, but that's not really what light therapy looked like. If you look at this actual picture of some men in their underwear undergoing heliotherapy from the National Library of Medicine, it's not quite as glamorous as a previous picture might make it out to be. So in the 1920s, Alexander Gurwitch showed that onion root meristems emit faint radiation capable of stimulating mitosis in adjacent tissues. He coined the term mitogenetic effect to describe light-induced cell replication and founded the study of biophotons. Now, this research we now know has been confirmed, at least insofar as we now know that all living organisms emit faint radiation, now called ultraweak photon emissions. They span the UV, visible, and infrared spectrum, and it's most well documented through the UVA to visible range. Now, UVC wavelengths, which are fairly high frequency, high energy photons, are also implicated in even promoting cell replication. And these are distinct from thermal light emissions. In fact, all UPE are distinct from thermal light emissions that you might view in an infrared camera or night vision goggles. Now, even though Gurwitch discovered biophotons, which have now been conclusively confirmed, he framed ultraweak photon emissions as a biological signaling mechanism. This work was studied and reproduced in the 1930s, but then fell into intense scrutiny and even skepticism. And today, Gurwich's ideas remain contentious, although his contribution is becoming increasingly acknowledged. As we continue our historical tour, it's worth noting that all of this theory of the interaction of light and life comes in the context of a long-standing debate between mechanicism and vitalism. which certainly goes back to the 1800s, at which time the so-called vitalists argued that a life force imbues living organisms with qualities that are absent from inanimate matter, in opposition to the purely mechanical view of life, as if we were all tin soldiers or wind-up toys. So by the 1930s, this vitalistic philosophy had been very successful. Both Niels Bohr and Henri Bergson by 1930 had won Nobel Prizes, and they rejected the idea that life admits an entirely classical mechanistic description. By 1948, Norbert Wiener, in his masterwork on cybernetics, argued that the stability in living matter arises from a high internal resonance, that is to say, isomorphic quantum state transformations, so as to make small causes produce appreciable and stable effects. So there was already some mathematical theory being developed to explain how life operates at all. And with some caveats, Wiener argued that vitalism has won in the debate to the extent that even mechanisms correspond to the time structure of vitalism. Now, what is that time structure of vitalism? Well, the idea is that in Newtonian mechanics, time is reversible. Everything is deterministically predicted. If I knew every particle's position and momentum in one moment in time, I could not only predict everything that would ever happen in the future, I could actually retrodict everything that had ever happened in the past. But people like Henri Bergson argued that life really doesn't operate that way at all. That living systems operate in a way that is not reversible, but innately irreversible. We all are born young and grow old and die, and life around us is ever changing. So Bergson, in his picture of the time structure of life, argued that the more we study the nature of time, the more we shall comprehend that duration means invention, the creation of forms, and the continual elaboration of the absolutely new. And so this uplifting kind of picture of life, not just as a mechanical thing, but an endlessly creative thing, inspired people like Bohr and other vitalists even into the mid-20th century. Now, Bohr, in his talk to the Society for Light Therapy, proposed a principle of complementarity for biology. And he questioned the theory that new biological forms arise purely through accidental disturbances to gene structure. Inspired by Bohr's lecture, Delbruck pursued experiments that bridged quantum theory and genetic change, ultimately winning the Nobel Prize in Physiology and Medicine in 1969. And this overall theoretical picture was developed, also motivated Schrodinger's 1943 lectures on What is Life, where he described a stable molecular code that defies thermal noise, precluding the likelihood that life could rely on the function of classical genetic material. And this informed research by Watson, Crick, and Wilkins, who of course won the Nobel Prize for discovering the helical structure of DNA, which they built on X-ray crystallographic data produced by Franklin and Gosling. And if you hadn't seen it before, here is the famous photo 51, the X-ray diffraction image, which inspired the helical model of DNA that we all know so well today. So Bohr wasn't done yet. He gave his 1932 talk, but 30 years later, Delbruck, who had made a name for himself by then, invited Bohr to deliver the opening lecture at the dedication of the Genetics Institute in Cologne, where Bohr revisited the theme of his talk to the Light Therapy Congress 30 years earlier. Now, that would prove to be Bohr's last public lecture before he died. But the story didn't end there either. By 1976, Delbruck completed the trilogy with a talk called Light and Life 3, in which he presented organisms as enormously complex molecular systems, and he proposed that with increased capabilities, humans could create technologies that might even surpass forms found in nature. And this goes beyond classical physics and photochemistry. Advancing photochemically active molecules is critical for life, Delbruck curated a list of photochemical biomolecules that play essential physiological roles and proposed the following fundamental topics. He discussed photoreceptors, chlorophyll, protochlorophyll, rhodopsin, phytochrome, cryptochrome, which of course contains flavin, and photolyase. He discussed quantum transfers, biomolecular photoexcitations, electron and proton transfers, and even conjugated molecular bridge systems such as beta-carotene. And he even mentioned ROS physiologies, reactive oxygen species that can act as key mediators, coupling photochemistry to cellular physiology, of course, along with the photosynthesizers that absorb light and then initiate those key photochemical reactions. So even in the 1970s, Delbruck was already discussing many of the fundamental topics that continue to be important in quantum biology today. So let's look at those frequencies of light. Different frequencies of light can have many different effects on health and disease in living cell. For example, infrared at wavelengths above 720 nanometers can protect against chronic disease, are proposed to improve mitochondrial function and ATP production, and have been used as an emerging therapeutic tool. Visible light, of course, impacts retinal function, sleep, and mental health. Artificial sources of light have been linked to oxidative stress, neurodegeneration, and forms of metabolic disease, suggesting that natural sunlight is indeed as healthy as it has often been purported to be. And ultraviolet wavelengths below 380 nanometers, of course, in excess can induce mutagenesis, accelerating aging, or even causing skin cancer, while moderate doses of ultraviolet light are beneficial. Wavelength-specific quantum effects may produce discrete biological outcomes. And in fact, we are seeing that more and more with the study of treatments like photobiomodulation, which we will discuss later in the book. And so if we look at, for example, these ultra-weak photon emissions that all living organisms that have been studied have been found to produce. As I mentioned, Gurwitch discovered these in the 1920s and 30s, but a lot of momentum on that research was lost until the 1990s, where experimental studies using photomultiplier tubes really conclusively confirmed UPE across various plant, animal, and human cells. And now UPE are confirmed in all living organisms that have been studied. They have intensities in the range of 10 to about 1,000 photons per centimeter seconds, and a spectrum of roughly 200 to 1,000 nanometers, although of course more study is needed in that area. Ultraweak photon emissions are produced wherever you see chemical reactions in the cell. They're primarily believed to be produced by mitochondria, but DNA has been proposed as a secondary source. Oxidative reactions and other reactions that produce reactive oxygen species are strongly implicated in the production of these weak photon emissions. They are implicated in optical coupling, even, for example, in the brain. Gurwitch proposed they had this mitogenetic effect, that UV range biophotons might even promote cell replication in the right amount. We certainly know that cells that are stressed tend to produce more UPE, and as a result, they have been proposed for use in medical diagnostics and health monitoring. So what's fascinating about this is that cells emit different intensities and even different spectra of UPE depending on their metabolic state. Are they healthy? Are they dividing? Are they stressed? Are they diseased? Since different cellular processes will naturally produce different photons, different spectra of photons, different oxidative reactions associated with different cell processes should result in different spectral bands. And a lot more work needs to be done in this area to see how far we can take UPE diagnostics to understand the interplay between health and disease in living cells. These have already been proposed as potential aids for early cancer detection, tumor diagnostics, and even observations of tumor destruction, that is necrosis, during chemotherapy. But they've also been proposed for use in agriculture to tell whether agricultural product is healthy or diseased, likewise in food and herb inspections, also neurological imaging, and of course, all kinds of health monitoring. Now, UPE also potentially have an interesting role in relation to the main structural element of the cell, the cytoskeleton. So UV light-induced microtubule structural dynamics, that is to say that UV light can change the dynamics of microtubules. They reorganize structurally under UV light, and this presents the possibility of photocontrol of biological function via structure photochemistry relationships. If you can use light to talk to the main structural component of the cell, that presents interesting options for controlling the structure of the cell, which of course strongly influences its function. We know that oxidative stress is associated with the production of these biological photon emissions by singlet oxygen and triplet carbonyls, for example, and that reactive oxygen species are formed during cellular metabolism. And of course, the dynamic properties of microtubules, for example, growth and shrinkage rates, rescue, and so-called catastrophe as the microtubule falls apart, are significantly affected by the oxidative environment of the cell. They're influenced by the presence of these reactive oxygen species. And so reactive oxygen species are considered as regulatory mediators which govern cytoskeletal dynamics, regulating microtubule formation and actin polymerization, as well as tubulin modifications, such as glutathione. And so the interplay between the cytoskeleton, reactive oxygen species, and various forms of ultraweak photon emissions poses interesting possibilities for photochemical control of cell processes. For example, white blood cells that were stimulated to undergo a respiratory burst with light could activate a second burst in a population of chemically separated but optically coupled white blood cells, obviously already demonstrating a simple proof of principle of optical communication between cells. And cancer cells exhibit unique photon signatures that are accompanied by profoundly disrupted redox chemistry and microtubule dynamics. And of course, it's worth mentioning that microtubule stabilizing drugs that limit microtubule dynamics, like taxanes, are among the most common clinically deployed chemotherapeutics. So this is not exactly conclusive, but it paints a picture that is strongly suggestive of an interplay between light, chemistry, and the various structural and chemical processes that go on inside the cell. If we consider the cytoskeleton as an optical integrator, in fact, research has shown that experimental work by Albert Buehler indicated that fibroblast cells can sense and respond to near infrared light, including light from other fibroblast cells, mediating long-range attraction between those cells. The centrosome near the nucleus of the cell has been proposed also by Albert Buehler as a cellular eye, and a perpendicular pair of centrioles, which make up the centrosome, have in that view all the geometric features of a cellular eye. So experimental evidence confirms that the mechanism for detecting infrared light sources is located in the fibroblast centrosome. Now this is interesting. This is optical processes at the center of the cell where you wouldn't obviously expect them to exist. Now, the cytoskeleton that we now know can respond to both far infrared and near field UV light. And so additional research that indicates that the cytoskeleton may also mediate nonlinear optical effects suggests that these signals might be combined to give the cell a sense of its optical surroundings that certainly warrants further investigation. I've proposed an open quantum systems theory of these kind of phenomena, and we really need to develop precise mechanistic links between metabolism, ROS regulation, UPE, and cytoskeletal dynamics. As I mentioned, I've argued that an open quantum systems theory is really necessary to model these effects. So this brings us to the idea of quantum biochemical control. Biophotons and condensates, what are those? Well, biophotons have been proposed in neurons. Myelinated axons in neurons have been proposed to act as photonic waveguides in the brain, linking quantum mechanics to neural signaling in work on quantum and biophotonic neural effects. Electrodynamic forces have been implicated as coherent excitations can enable long-range electrodynamic forces between biomolecules such as BSA, bovine serum albumin, which have also been proposed as a key mechanism for focusing thermal energy in protons during enzymatic reactions, for example. And additional ex vivo studies have shown strong light matter coupling in biological molecules can generate condensates of organic polaritons that can also influence chemical reactions and collective dynamics. So these implications for electrodynamic forces, biophotons, and even quantum mechanical condensates, like similar to Bose-Einstein condensates, these polariton condensates, suggest good reason to pursue further research at the intersection of quantum optics and quantum biology. Investigations have shown signatures of exciton delocalization and superradiance in bacterial photosynthetic systems, where it's indicated that the high efficiency of photosynthetic light harvesting may result in part from excitonic quantum coherent effects in photosynthetic antenna complexes. Light harvesting in aromatic networks have been implicated as protein polymers like microtubules and actin filaments have been found to exhibit interesting light harvesting characteristics in and of themselves without dedicated light harvesting chromophores. So for example, the cytoskeleton can respond to UV and infrared light. Studies have shown that microtubules have the properties of good light harvesters. And we know now that there are even photoreceptor proteins like UVR8 that I discussed previously that rely on tryptophan networks for light harvesting or light reception alone. And so this really does strongly indicate that microtubules and perhaps actin filaments, the cytoskeleton in general, have optical properties that are important for its function, but yet to be determined and fully characterized. In my own work, we found that microtubule superradiance was indicated by fluorescent quantum yields in our simulations and experimental work, and this suggests that coherent collective excitations in these systems may play important functional roles in aromatic networks, for example, tryptophan networks found in microtubule and other architectures in the cell. So this really presents the importance of studying quantum optical phenomena in biology. Strong light matter coupling can overcome quenching in artificial carbon nanotubes similar to biological microtubules, where analogous brightening effects were shown in the lowest exciton state. Self-aggregated nanotubes of chlorophyll can support macroscopic coherent states even at room temperatures, which also has implication for light harvesting and energy transport. And if you're wondering, well, how could these kind of quantum optical effects exist in biology? Don't we need cavities, mirrors and ways of trapping light like we do in a laboratory? Well, not quite, because in fact, ultra-strong light matter coupling was demonstrated in simple systems of organic dye molecules embedded in dielectric films. And so this really shows that the conventional tools of the quantum optics laboratory, lasers and various mirrors and high quality confinement of light, may not be necessary in order to investigate different kinds of strong light matter coupling. And other important and intriguing results have demonstrated, for example, that chlorophyll A can exhibit electrodynamic behavior when placed in a plasmonic nanocavity. And in another result shown artistically here, quantum entanglement of a microscopic tardigrade, a water bear, was demonstrated when the water bear was put in cryogenic hibernation, echoing Bohr's principle of biological complementarity. Bohr's idea that we cannot properly interrogate the physical properties and the living properties of a living organism at the same time. Of course, the figurative image shown here is not a true picture of the experiment with the water bear, but what happened in that experiment with the water bear, which can go into a cryogenic state, a state of deep hibernation, was brought down to cryogenic temperatures, entangled with a qubit in a quantum information experiment, and then warmed up and brought back to life. So these kind of experiments really capture the imagination, not just of quantum effects in biology, but of curious ways of interrogating life with quantum optic techniques. So in conclusion, I'd like to suggest that all of this presents interesting opportunities for photochemical control of biological processes. There are exciting therapeutic prospects as new photochemical therapies may target microtubule dynamics in cancer cells and neurodegeneration. Ultraweak photon emissions are already being studied as a non-invasive biomarker for metabolic cell states and cellular stress, with important implications for diagnostics. We've discussed the potential for cytoskeletal control. Light-induced microtubule stabilization and cytoskeletal morphology control have been demonstrated experimentally, and results of these kinds have great potential for further work in embryology, regenerative medicine, and immunology, just to name a few. Now, this also is likely to impact the development of new types of biotechnologies and biophotonics. For example, advances in research at the intersection of biophotonics and quantum optics are likely to lead to new technologies, including organic laser systems, perhaps super radiant or polaritonic lasers, and also the potential for interesting biotechnologies developed using engineered biomaterials, quite unlike many of the materials used in quantum optics today. So if you found this fascinating, please don't hesitate to download your guide to quantum biology, Physical Principles of Quantum Biology. You can get it now today from the Physics Archive. I hope it will attract the attention of researchers, students, and other curious minds ready to venture beyond the limits of classical biological explanations. And if you have any further questions, don't hesitate to email me at my email address. You can continue to watch these YouTube videos on my YouTube channels or check out my website. So I hope you've enjoyed today's lecture and have a great day.